Okay, so the last portion of the lecture, we're going to discuss the functional anatomy of the photoreceptors and how you accommodate your vision for light and dark vision. So the rods and cones are modified neurons. So you have the receptive fields that are going to sense light that is coming into the eye. So the light waves will hit these receptors. They will then send a signal to the bipolar cells and then the ganglion cells, which will create a signal within the brain that interprets images that you see. So the receptive regions are called the outer segments, and so they contain the visual pigments, which are photopigments, and these are molecules that change shape as you absorb light. And different wavelengths will create a different signal or cause different neurons, different rods and cones to be to depolarize, which essentially sends that signal. Because if they depolarize because of a certain wavelength, they will activate neurotransmitter release to the bipolar cells. So the inner segment of each will join the cell body. So different regions that are going to have different purposes. So the outer segment is going to contain the visual pigments. So the photoreceptor cells are very vulnerable to damage. They are very, very sensitive. So if you're looking at any sort of extremely bright light, or if there's a sudden change, it can lead to significant damage. That's why you don't look at directly at an eclipse, and that's why you don't look directly into bright lights for any length of time. There is degeneration if the retina is detached, and that can be from a loss of intraocular pressure, loss of vascularity to the choroid region, and there are several other options as well. But it's also destroyed by intense light, so you don't stare directly at intense lights for long periods of time because that can damage your photoreceptor cells. The outer segment is renewed every 24 hours, so you're constantly replacing your rods and cones so that you don't have destroyed or damaged rods and cones that are getting improper visions. So the tips will fragment off and then become phagocytized so that they are damaged, removed, and replaced. The rods, those are very sensitive to light, and they're best suited for night vision and peripheral vision. Okay, so when you wake up in the middle of the night and you're trying to find your way to the bathroom, the rods are going to be the ones that are activated, and they will produce that gray tones. They also are the best for peripheral vision. So if you notice something in the background or off to your sides that has sudden movement, that's when the rods are activated. So they're very sensitive to light, but they don't see clearly. So you're not going to have a distinct image. You just might see something flashing in your peripheral. They contain a single pigment, so they don't get colors. It's only perceived in gray tones. So at night, that's why you don't see any good colors, because you don't have enough light to activate those cones, so therefore you get gray tones. You're essentially seen in black and white. The pathways will converge. They cause fuzzy, indistinct images. So something that you're not focusing on in your periphery is going to have those fuzzy, distinct images. It's difficult to be able to focus on that, but there are times where, if you've ever seen the show, it's called Brain Games, they will actually talk about these issues and have fun experiments that you can do to see the difference between your rods and your cones. But if you're looking at something in your focal point, that's going to be mostly the cones, so you get that HD high-res image, whereas anything in the periphery is going to be indistinct. So the cones are going to need bright light for activation. So in low light, you aren't going to be able to see in color. So you need the bright lights around in order to activate the cone to get that high intensity image. They do react more quickly, so they adapt early and often. They have one or three pigments for colored view, one of three, I'm sorry. So there's three different cones that each individually will absorb one wavelength of light. And so you combine them together with the rods to get specific colors. These are non-converging pathways, so each cone will go to one bipolar cell, and that way you get a single distinct signal. It's like increasing the amount of pixels that you have in the screen. So instead of the 820, you get the 1040. So that's what we're looking for. Color blindness is the lack of one or more of the cone pigments. So if you're just lacking one of them, you might not realize that you have color blindness immediately. You can do some tests to figure out if you are lacking one of the cones. Anybody who's lacking all three images, they would only see in gray tones, have blurry, indistinct vision. So this is a good chart to tell the difference between the rods and cones. So I would really study this and be able to create your own charts between the two. Understand where you're going to find cones, where you're going to find rods, how they are activated, which one sees color, which one sees the high definition. The cones are going to be essentially the advanced version of the rods, because they pick up colors, they get that distinct image, and then there's only a 1-to-1 ratio. So light adaptation. When you're moving from a dark room into bright light, okay, so let's say you wake up in the morning and you have all of your shades drawn, it's pitch black, and then you open your shades and now it's bright light. What happens? How do you see? Well, both rods and cones are strongly simulated at that point. Your pupils are going to start to contract and constrict because you don't want to have that much sunlight coming in all at once. Otherwise, that can be kind of blinding. That can be damaging. So large amounts of pigments are broken down instantaneously, which is what produces your glare, so that you have all of these pigments that are just sitting there getting in the way. Visual acuity will improve over 5 to 10 minutes. So the rod system will start to shut down while your cones turn on. And the cones are rapidly adapting, they're rapidly turning on, so this is a relatively short period of time to get your full HD image picture. So the retinal sensitivity will decrease as you begin to get used to the light, and as the rods are turned off more and more, so that you're getting that high-res image. So cones are rapidly adapting. You get 5 to 10 minutes that you're going to be able to see properly without having to squint and shield your eyes. Dark adaptation is going to be slightly different, because if you're moving from a bright light into pure darkness, so let's say you are in a room and you turn off all of the lights and now you're trying to find your way out. Well, is it easy to do? Well, no, because it takes a while to shut off the cones and turn on the rods. So the cones stop functioning instantaneously because they don't have enough light to activate them. You need bright enough light to be able to increase the signal. So the rod pigments are bleached, the system is turned off, and you need to turn on that system. So rhodopsin will accumulate in the dark, transducin returns to the outer segments, which is the light sensing segments. The retinal sensitivity increases within 20 to 30 minutes, and the pupils are dilating because you're trying to get in as much light as you possibly can. Okay, when you have a lot of light around, your pupils will be relatively constricted. So as they start to open up, you get more light, which will actually go to the rods rather than just the fovea centralis. So you're not hitting the cones that aren't going to adapt to the very small amount of light that you have. You need to get the lights on the periphery where more of the rods are. And so that takes a little time, and it takes a while for the cones to adapt. This is going to be like the old analog TVs that are the black and white. So again, it takes a while for those to warm up, to turn on. I don't know if you've ever had those TVs, but you turn it on and it starts to make a little noise, and it kind of whirs, and then the TV finally turns on. Okay. So night blindness is known as nyctalopia. Okay, opia is going to be for the eyes and for vision, and nycta would be for night. So night blindness. This is when you have degeneration of the rods, because the cones are going to be turned off. Okay, when you have low light. So when you don't have enough rods, when they have been degenerated, that would lead to the inability to see in low light. Okay, that would also lead to problems with peripheral vision. So this is commonly caused by vitamin A deficiency. Now, in the United States, that is typically not much of an issue. In some of the developing countries, it is more of a problem with having the vitamin A deficiency, because we have vitamin A put in practically everything. So if administered early, vitamin A supplements can restore the function. You can restore the rods. But if it's not caught until those rods are pretty much all destroyed, well, there's not a whole lot of reversal. Okay, also caused by retinitis pigmentosa. So retinitis, inflammation of the retina. Okay, it's a degenerative retinal disease that will destroy the rods. So it's a progressive chronic inflammatory degeneration of your rods. So how do we get the signal from the eyes to the brain? Well, the axons of the retinal ganglion cells will form the optic nerve at the optic disc, which is your blind spot, and they will send signals to the brain, but there's several pathways that they can take. Some will go to the optic or occipital lobe, but there's other regions of the brain that will also play a role in sensing vision. So the medial fibers of the optic nerve will decussate at the optic chiasm. Now the decussation is going to be it crosses. So only the medial fibers, so the medial half of your eye, will cross and go to the opposite lobe, while the lateral fibers of each eye will stay on the same side. And you're asking yourself, why does that happen? It's because it's the best option to create the depth perception. If you only have one eye, you won't be able to tell how far something is away. So the more you can share that visual field, the better. Now, most fibers of the optic tracts will continue to the lateral geniculate body of the thalamus, and the thalamus is going to send the neurons to their proper destination. Okay, because that's what the thalamus does. That it essentially is a packaging center that sends the neurons where they need to go. The fibers from the thalamic neurons form the optic radiation, so they will radiate out from the thalamus, project to the primary visual cortex and the occipital lobes, where you will create an image in your brain. So fibers from the thalamic neurons will form the optic radiation. The optic radiation fibers connect to the primary visual cortex, and then they will also communicate with the association centers. Other optic tract fibers will send branches to the midbrain, which would include the superior colliculi, which initiates the visual reflexes. Now that would be constriction and dilation of pupils, but the inferior colliculus would be for the hearing, the superior would be for vision. So we're going to be coming back to that. So there is also a small subset of ganglion cells in the retina that contain melanopsin, which is part of the circadian pigment. That projects to the pretectile nuclei, which is involved with pupillary reflexes, and the suprachiasmatic nucleus of the hypothalamus, which is the timer for daily biorhythms. So essentially, when you have sunlight, you are going to stimulate your body's awake cycle. And when you have this dark, when it starts to get dark out, your body has that rhythm to kind of lull you to sleep. You're trying to your body's trying to get you to sleep when it's dark and stay awake when it's light. So when we are using things like our computers or phones or TVs late at night when it's getting dark out, when we're laying in bed, that messes with our circadian rhythm. So your daily biorhythms are thrown off because your body now thinks that it is day out instead of it being nighttime. So your body is still in that awake mode when you have those lights on. So there's different options. Some of the most popular ones are: don't put a TV in your room, don't bring your computer in your room, charge your phone outside of your room at night. There are also blue light glasses that you can wear at night when you're looking at your phone, so that now you don't have the you don't have the blue light that will activate this reflex that keeps you awake. So there's several different options when it comes to the visual pathways in the circadian rhythms. Okay, so this is a chart that shows you where the fibers will go. So you have the right eye, and the lateral portion will go to the right occipital lobe, but the medial portion is going to go and decussate at the optic chiasm right here. It's going to go through the thalamus, where it's going to go to this optic radiation, so they radiate out like sunbeams. Okay, but some will also go into the lateral geniculate and to the superior colliculus, which are going to be for the circadian rhythms, but also for some of the reflexes. So again, you have this fixation point, and the fact that you have half decussating and half staying on the same side, you're going to get more of that the depth perception. Okay, so both eyes view the same image from slightly different angles, and depth perception results from the cortical fusion of slightly different images. So your left and your right side will see things just a little bit differently, and your brain can interpret that as determining how far away something is. So it does require input from both eyes. So if you only have one eye that you can look out, so if you cover one eye with a patch, or one eye does not function very well, you don't have very good depth perception. If you are trying to look through the barrel of a rifle, for example, during hunting season, that might not give you good depth perception, but it can improve your accuracy. So visual processing is going to occur in the lateral geniculate nuclei of the thalamus, and that's a process for depth perception. Cone input is emphasized here, so the HD image, and your contrast is sharpened, so you get the clearest picture that you possibly can. The primary visual cortex will have neurons that respond to dark and bright edges and object orientation. Provides form, color, motion inputs to the visual association area, so the prestriate cortices. Okay, so form, color, motion. This allows you to see images in what they should be appearing in the proper orientation. So this gives you a good review of where you need to be studying. So knowing the three different layers of the eye: the fibrous layer, the vascular layer, the inner layer. Okay, know those three different layers of the eye and be able to tell me what components are in each of those layers. Okay, so fibrous is the sclera, which is in the posterior, and the cornea, which is the anterior, where you bend the most light. The vascular layer is the choroid, which supplies blood to the retina. The ciliary bodies, which is going to be attached to the lens via the suspensory ligaments. And then you have the iris, which is going to be the portion of the eye that gives you the color, so blue, hazel, all that. And then you have the inner layer, which is going to be the retinal layer, and that's only in the posterior region. Okay, tell me the difference between the rods and the cones. Go back through those charts, be able to create your own. Which one is the HD image? Which one does color? Which one is found in the fovea centralis? Which one is going to be for light adaptation, dark adaptation? Okay. Now you go through, the light is going to pass the ganglion and bipolar cells, and then it's going to activate the photoreceptors, which send a signal backwards from the photoreceptors to the bipolar, to the ganglion, and then out. Okay. Tell me about the difference between the posterior and anterior segments. So the vitreous humor, where is that found? When is it created? Does it get created constantly? Is it lasting a lifetime? Then the anterior segment. Okay, what produces this aqueous humor? What drains the aqueous humor? Okay, where does the anterior and posterior chamber separate? Then tell me how light passes into the eye. So it goes through the cornea, the aqueous humor, the pupil, the lens, the vitreous humor, then the retina, where it's read by the photoreceptors, the rods and the cones. Okay, so the lens. How do you adapt the lens for close vision? Okay, well, how do you adapt for close vision in general? What are the three components of that? Okay, tell me how the the lens will work with close vision and with far vision. Okay, light is only seen in the visible light, so those are wavelengths of light, and they're read by the rods, which give you the gray tones, the blue, the green, and the red cones, which combined will give you the colors of the rainbow. And knowing how concave and convex lenses are going to alter your visual field. Okay, so if somebody's nearsighted, do they need a concave or a convex lens? How do you focus? Okay, at what distance do you no longer have to adapt your lens to be able to see the same image? Okay, so close vision, accommodation of the lens. Okay, your convergence of the eye, and then it's going to be constriction of the pupils. Okay, how do you adapt to light and how do you adapt to dark? I'm going to ask a question about that, so you need to know at least a couple of different things about light and dark adaptation. So when you're going from a dark to a light room, which is turned on, rods or cones, and which one is turned off, rods or cones? Okay, how long does it take? Is it fast or is it slow? Tell me those two things about each of those and you're going to be pretty safe. The pathway in the brain. Okay, so those are the main points that are going to be on the exam on Thursday. So make sure that you get all that information down pat.